What I'm going to try and do is sort of give the lay of the land with respect to measurement needs, talk a bit about some thoughts I've had and issues that maybe you all haven't considered so much when it comes to measurements, and hopefully develop some conversations about what the real measurement needs might be. I have always liked this quotation, "To measure is to know," from Kelvin. But the question is, well, what are we supposed to be measuring? And then, of course, if I measure these things, will it make us safer? Are they the right measurements? So we've heard a lot about particle size, particle shape distribution, particle surface chemistry in solution. What solutions? Well, that's an interesting question. Clearly, and maybe this one ought to be at the top, interaction of particles with biological systems, toxicity studies, and the key here, I think, what I'd like to point out is well-characterized system. That is, materials who have some pedigree. If we're doing interlaboratory studies, that everybody's doing it on the same material. There's been a lot of discussion over many workshops and so forth of databases. That is, once we learn something, how do we put it in a format so that others can use it readily, can exchange their data on whatever property they're measuring with me, or I can exchange with them. And so the databases and data exchange formats, that can be a very daunting task. Measurement guides, measurement protocols and guides for the non-expert. I personally like this a great deal. We have to recognize that many of the people who are and will work in this field are not expert chemists. They are not PhDs. They need to measure things, but they need to understand what the pluses and minuses of a technique and where things can go wrong. I think modeling techniques so as to be able to predict what we think will happen. There are too many experiments to be done to do them all. We need some guidance in the beginning to tell us where we should go, what's going to be critical, and what we should expect. There was a plea made that if I'm a small company or I need to make measurements outside of my laboratory, I need portable, inexpensive instrumentation so I can get these measurements done easily. There was some mention made early on in this workshop that there's an awful lot of nanoparticles out there. There are diesel emissions and so forth. And the question is, how do we detect what we call man-made nanoparticles against a background of all of these other nanoparticles? I mentioned this idea of having something now for the non-expert. And this is simply one representative idea of what at NIST is called a practice guide, but other organizations like the National Physical Laboratory in Britain have similar kinds of things. I'm a great believer in the idea that we need to tell people what we know now, even though we are not, in fact, we're sure we don't know everything that needs to be known to do this, but it's the best we can do now, and here's today how we think you should make these measurements. But I believe, after saying all this is kind of in a rough idea where we stand now. But I see a couple of problems that I'd like to talk about with you, and hopefully we'll get some discussion on. First, as I mentioned earlier, are the measurements we make on these inorganic particles or nanotubes or quantum dots really going to lead to an understanding of the hazard? And second one that I will talk about is, what is the role of measurement uncertainty when we say we have a risk with respect to these materials? There are lots of efforts going on to develop measurement technology. There's no question there. Both as formal and informal standards, the world is concerned about safety of nanomaterials, and there's a large effort being put into doing this. I think that when we do this, that we better be sure we know how well we can make the measurements and what the uncertainty bands, what the standard deviations are, the real standard deviations are, and so forth are in making this and determining the risk of any of the materials we're looking at. Obviously, I said it earlier that one of the critical parameters, but I come back to it because I think it's so important, is that detailed knowledge of the reaction mechanisms between nanomaterials and tissue are going to be critical to developing safe procedures. I certainly believe that better computation and modeling techniques are going to be needed to accelerate the process of development of safe materials and accelerate the development of the measurements themselves. Again, in the same light, this idea of going from in vitro to in vivo as quickly as possible, we really don't know. I mean, I'm convinced from my looking at what happens with crystalline silica that we can make all the measurements we want on the outside and characterize the heck out of those particles, and we will not be able to tell why one particle is harmful and the other one is not. So I strongly suggest this.
I'm going to tell you about the building blocks. Some of this you've already heard, but I don't think it's problematic to repeat it. And I'll try to identify some of the important features and why a holistic evaluation is essential. And then, of course, the main part of the talk where we want to look at controlled synthesis to provide materials. And so what if I can make fancy materials? What is the fate, even while doing let's say, a toxicological test? And I'll try to identify that. And we all work in labs where we make these things. Are our labs safe in the first place itself? So I feel that we need to look at the system holistically. It starts right from a synthesis process, maybe an occupational environment. You want to trap these particles. If they are harmful also, we want to trap them. If they are useful, of course, I want to put them to use, so I want to collect them. Then comes the issue of handling. It's not only collecting it right here. I have to really handle them and go and make a product. So there's the aspect of handling and processing, and then eventual use of the final product that we make. And all these, of course, relate to exposure. At least we have the nomenclature right. And then, of course, we have things like the dimension of the size. How do I characterize something like this? I can use a fractal dimension. I can use a radius of gyration, a mobility size. I bring up mobility size because we design instruments to measure these in flight. I might be interested in the surface area. There are ways now to interpret the surface area by light scattering measurements and all kinds of different measurements. And, of course, eventually the shape and maybe the fractal dimension sort of describes the particle shape.
So some common misconceptions here I point out. Here, for example, such as all nanoparticles generically behave the same way, not only for inhalation, but all of them, as I said, as showed in one of the headlines before, are toxic. And you need to be very careful. The smaller the nanoparticles, the deeper they deposit in the lung when they are inhaled. All nanoparticles translocate rapidly across barriers, epithelial barriers along nerves, and get into all different tissues. The biodistribution of intravenously administered nanoparticles is the same as that following delivery to the respiratory tract. Just to make it easy for people to think that if they do an IV study, look at the biodistribution, that's what they also should expect from inhalation or administered to the lung. In vitro studies are sufficient to identify toxic nanoparticles. Yes, they can, but we have to be very careful how to do that. I will get into this here. And finally, in vitro tests are predictive for in vivo responses. Again, it's a very difficult and involved issue, and I try to explain that a little bit more when we go along. And once they have deposited, they distribute throughout the body. They show the scheme here with the uptake into skin, respiratory tract, GI tract, or also drug delivery to the blood. And you see that the blood seems to be a very nice plenum to distribute it throughout the body here. And David Granger this morning pointed that already out, that indeed it goes to everywhere. Most efficiently, of course, will be caught in the RS organs, liver, kidney, and spleen. But basically it goes to everywhere, including the central nervous system also. Now, one aspect we need to be aware of is that these translocation rates are very, very low. And I show you also some data, as did David this morning, that indeed we should not assume that all the particles deposit in here upon inhalation, and I will focus in my talk on the inhalation route of exposure, that they will quickly get across and distribute then rapidly throughout the body. Now, we have heard quite a bit of this already, physical chemical properties, and how important they are for toxicology, for their effects here, so I don't need to dwell on these for long. Certainly size is of importance, but clearly the surface properties are the most important factors that we have to consider here. And here a question came up also today, surface area, how to measure that. And BET surface area is an easy thing, relatively speaking, easy thing to measure. But is that really something that is equivalent to a biological available surface area? I don't think so. So we probably should try to find out what is a biologically relevant surface area if we use that as a dose metric. Key parameter, of course, for all of these is the dose, and that's what I will mainly focus on. So inhalation is really the gold standard, and to determine effects of airborne nanoparticles, obtaining also regional deposited doses, which would be important when you target certain cells, establishing the biokinetics of materials that you are interested in, and in general then provide a guide for in vitro dosing of respiratory tract cells. So just closing with some considerations for design interpretation of toxicity assays. Certainly, as has been pointed out several times here, key is the physical-chemical characteristics here, and of different ones. We should not going overboard with this, but it's very important to at least to look at the key parameters here, and the in vivo biokinetic data to serve as a guide for in vitro dosing and in order to verify the steady dose. And that is very important to use a wide range of doses, including realistic relevant doses. So I'm not saying that these high-dose studies are necessarily meaningless, but it's the way to how they are interpreted that can often be questioned as being meaningful at all. So we should use a wide range of doses. Dose weight clearly is a potential confounder, and the impact of the modified surface area. I mentioned that before, the use of dispersant in vitro studies, also in vivo studies, when you instill, you use dispersant. How does it affect the outcome? We don't know yet for sure. Clearly, the in vivo validation of any in vitro system with relevant target cells should be done, should be confirmed. And then the analysis of the dose-response relationships for this extrapolation process, I think, should be closer looked at, and rather than just simply using a specific response, certain cytokine or whatever, being expressed, being activated, that may not do it. And finally, then, to characterize the hazard based on the surface reactivity. Let me that's a very important issue also, the extrapolation from acute to chronic responses. And that is a very important one by necessity. Of course, in vitro studies are acute studies only, and to extrapolate then from that to a chronic situation requires some more research evaluation. So I close with a series here of a general paradigm here, or concept here from in vivo studies to human exposures here. And clearly what is needed eventually to get some information about workplace exposures, whether it's in the laboratory consumer or at the workplace in the manufacturing side. And in vivo studies, the biokinetics are a main thing to look into, and the assessment of dose response, as well as the in vitro studies to select the appropriate target cells or tissues and look at the dose-response relationship with wide doses, so to then try to validate these studies here with in vivo results in terms of being able to predict it, and similar here from in vivo to the in vivo exposures here, trying to predict or to evaluate, to interpret the studies, and that we can predict what's going on here. Dosimetry is a very important issue here, and eventually we should be able to do just this step here. We cannot, of course, in perpetuum always do animal studies, as I pointed out before, so that would be a goal to do this. And once we can do this here, we can then also do hazard identification here based on what's going in here, maybe using the hazard scale that I suggested to use, and together with the exposure assessment, then we'll eventually be able to do some risk assessment as well.